Who wants to live forever? I mean, maybe not forever, but maybe a bit longer than actually expected. Today, we will learn some of the prerequisites to get there. Our next speaker, Moritz, will show us how AI and LLMs can help us to understand and manipulate biological cells. Moritz is a bioinformatician and machine learning scientist. He's doing postdoctoral research at the AI Institute of the Medical University of Vienna and CEMM. So thanks for being here, Moritz. And have a big applause for Moritz and his talk. Have fun.
Thank you very much for the kind introduction, and also thank you all for coming to this talk. It's really fantastic to stand on the stage here once. I've been several times at the Congress, and it's really something I'm looking forward to throughout the year. Also, before I kind of start, I would like to apologize for using this horrible corporate buzzword term AI. I use this interchangeably with machine learning and deep learning. All right, let's get started. And to kind of set the stage and bring some intuition of what's possible today, I would like to make a small quiz with you. So I tell you 3 ways we can and do maybe hack cells. And you can guess whether they're easy or challenging or maybe not even possible. So the first one are glowing animals. So what you see here is a picture of 6 mice, and 3 of them glow under UV light. Now, make up your mind. Do you think this is easy or challenging or even feasible? It's in fact relatively, of course, easy, and biologists are doing this for decades. The way it works is you introduce into an organism, such as these mice, an additional gene, and this gene codes for a protein. The protein is called, or the gene is called GFP, green fluorescent protein, and it has the function that it glows under UV light. All right, second one, using cells as biocomputers. So imagine you have a cell, and you can give it an input signal in form of chemicals, and then it does a computation for you. In this example here, maybe it can compute whether your input is a prime number, and then it outputs yes or no via changing its color or so. Do you think this is easy or hard? This is feasible, possible, but actually relatively challenging. And the way that biologists implement this is that they use, first of all, a relatively simple organism such as bacteria, and they introduce very well understood, very well controllable genes in there and configure and link them in a way that follows kind of Boolean logic, really. So they imitate a program written in Boolean logic within biology. And the last one is a gene effect prediction. So imagine you have a cell here, which is depicted here as a microscopy image, and you wonder, if I now delete 1 or 2 of its genes, what happens to the cell? Like, does it die? Does it proliferate faster? Does it become more active in something? And this is actually a quite challenging task. In many cases, this is unsolved and something that people are actively working on right now. And with this, I'd like to quickly provide an overview of what I will talk about. First of all, I want to provide some intuition of what cells are and how complex they are, actually. And here you see this microscopy, actually live microscopy image of an immune cell in yellow-orange. And what the cell here is doing is attacking a cancer cell and actually killing it. Because one important function of immune cells is clearing your body all the time from cells that become cancerous. And this is really just one example of the many, many kind of diverse functions that all the cells in your body have. You have cells to see, of course, like nerve cells, to think, cells to feel, to digest, etc. Now, in the second part, we then also talk how we can decode the cells with AI, like how can we understand their function, their biological functions and their identities. And then also, in the third part, how can we use this knowledge to also hack these cells and manipulate them in ways that help us. So cells are not only diverse, they're also immensely complex. So if we zoom in into a single cell, what we see is, even though this was only a tiny dot under the microscope, in fact, inside is like a whole kind of universe of different types of structures and different types of molecules, and they all have their different functions and interact in all weird ways to make the functionality of the cell possible. And this, of course, is an artistic representation that kind of only contains the molecular parts in the cell that we understand. In fact, reality is very different, and in reality, the cell is packed very densely with molecules, and much, much more is going on than we see here. And I encourage everyone to check out these beautiful videos on YouTube. They're called Cellscape. Now, if we zoom in one step further, we see that these structures that we just saw, they contain most of the times, or many of them contain of proteins. And we have 20,000 different types of these proteins in our bodies. And here's one that all of you have heard of, hemoglobin. It's responsible for carrying oxygen. So I just show it to you as an example of an average-sized protein. And to give you also more intuition of its size, this one consists of 10,000 atoms. So you see that if we zoom so much into the cell, then we really end up at atomic level, and this is where most of the biological functions are carried out. These proteins are built from your genes on your genome, which is made out of DNA. And the way that your cells are building these proteins is via an intermediary stage called the messenger RNA. That's also what is used in the recent COVID vaccines. And this kind of mechanism of first transcribing genes into mRNA and then into proteins is a very useful mechanism that cells also use to enable and disable individual genes in individual cells. And this way, cells manage to have all these different functions because they can enable and disable different genes. And conveniently, we can also use this mechanism to look at the state of cells. So what we can do is, if we have an individual cell, we can extract the mRNA present in this cell and look at it with a very specialized microscope called a sequencer. And what the sequencer can do is it can decode all mRNAs in the cell, and then if we aggregate this information with some bioinformatics analysis, we can get a measure for all the genes in our genome and whether they are on and off in the cell. And not only this, it's not binary actually, it's continuous values. So the activation of individual genes is not like on or off, but it's on a continuous scale. Some are more and some are less active. Now, a multi-billion-dollar industry has grown around this sequencing technology, and by now researchers use this kind of experiment to study cells by looking at the mRNA in many experiments, and they upload the results to public databases. So a lot of studies and a lot of cell states are available and protocoled online. There's, however, one catch here, which comes back to the complexity of cells. We still struggle with understanding what these measurements mean. And the reason that we cannot decode this so easily is that biology and genes and how they interact is super complex. So complex that this guy here famously said that nothing in biology makes sense except in the light of evolution. Now, to understand what he means by that, let's kind of take an example from software development. If you would develop maybe an operating system or a piece of software, you would engineer individual modules that have like very specific and clearly defined functions, and then you also you kind of engineer the interactions between them, and it's very well defined. Now, biology is kind of the complete opposite, because we heard like a couple of hours ago already a really beautiful talk about evolution in the glitch room, because evolution is based on random mutations, and then what you end up with is a system that looks more or less like this. You still have kind of these modules, which are genes, but they are highly redundant, and they have always a lot of functions like that are seemingly unrelated. Gene A could have 7 different functions which have no role with each other. And this, to us, it looks like a really completely chaotic system, even though in reality it's much more robust and efficient than you would imagine. Now, a quick recap of all this molecular biology that we're talking about in this introduction. So cells are extremely diverse and extremely complex. And they achieve this diversity via enabling and disabling genes, and we can also read out which genes are active in a cell by looking at these mRNA that they use to activate genes. All right, but as I said, still we struggle with understanding the states or the functions of individual cells. Now, how can we use AI to decode and interpret these cellular measurements? One of the core technologies that we are using are multimodal large language models. And multimodal here refers to the fact that the language model not only understands text, but also an additional modality. And in our case, this is or like in this example at least for now, these are images. So here I show you an example of ChatGPT, and I ask it this simple question: what does this image show? And then ChatGPT identifies it as a human figure leaping forward. I mean, of course, who doesn't see this orange figure running there? Let's try. But then if we try once again, oh, now it identifies it as the logo of an Indian stadium. If you Google the stadium, this is the logo. Okay, scratch that. Thanks for nothing, ChatGPT. At least the competition got it right. And I have my example for you on what multimodal large language models do. Now, you can imagine we can use the same technology to decode and understand cells. So imagine this scenario: we provide again a cell to the language model and ask it a question of what the cell is or what its biological function is. And then the large language model can answer. And thereby we have a system that really decodes these complex measurements that we struggle to understand into into our language, essentially. And to make sure that we all understand what we're using here, it's not a microscopy image that we would pass, but rather these gene activity measurements based on mRNA, because they're much richer in terms of describing the state of cells. Now, to understand this whole system, how it really works, we only need to answer 2 questions. First of all, how does this multimodal large language model architecture work? And second of all, what data do we train it with? So let's start with the first one. Of course, the core of this multimodal large language model is an ordinary large language model. And just very briefly how these language models work, especially in the generative settings when they generate text, like chatbots, like ChatGPT and other models, is the following. So the input text is always passed to them as a sequence of tokens or words in this case. For example, if you have what is the meaning of life, question mark, then the task of the model would be to generate an answer, of course, and it could be the meaning of life is 42 or so. And then what the model actually does is with this input sequence is it uses all its different layers inside and kind of makes an internal representation that allows it to predict a new token. So it outputs one new token. And then this token is appended to the input sequence again, and this process repeats. And thereby you have an iterative procedure in which you can generate new text. That's your ordinary AI language model assistant. Now, you need to know that in our instance, we used an open source language model because we wanted to manipulate this model called Mistral, the 7 billion version. And this model represents the individual tokens as floating point vectors of dimensionality 4096. So internally, it doesn't manipulate character sequences, but actually floating point vectors. Now, how can we introduce the additional modality of cell measurements in there? The idea is relatively simple. So, mouse pointer works, that's great. So we introduce an additional neural network. I call this here a cell model that is responsible for converting our gene activity measurements into abstract token representations that are in the dimensionality of 4096 dimensions. And this could be 2 or 3 or 4 or only 1, how many you want. And the model that we use here in reality is called GeneFormer. It's published last year. And this model is already really good in converting these raw gene activity measurements into a semantic representation of cell states and functions that can be used for downstream processing, as in this scenario. Of course, this won't work in this state because these two models are not aligned. They have different ideas of what these representations here mean. So we need to align them. And we do this by fine-tuning with a special protocol called instruction tuning. Now, how does instruction tuning work? The idea is the following. We give the model as input first this kind of measurement of a cell, and then also an instruction or a question. In this case, for example, what does this cell do? And then the fine-tuning signal, what we task the model to predict as a response, is a corresponding answer. And as you remember, this kind of cell is an immune cell, so a correct answer would be here. This is an immune cell, which maybe is killing a cancer cell. Now, the only question remaining, or maybe I can also add that this kind of enforces that the model learns to understand these additional abstract cell representations, because otherwise it wouldn't be able to come up with the correct response, right? Now, the only problem left is essentially to get training data. And as you probably know, these kind of models, they need a lot of training data. So we can't just ask biologists in our labs to kind of carefully analyze such data and come up creatively with conversations. Instead, what we did is we again relied on language model technology. And we did this on the basis of large-scale databases of cell measurements. I already told you a couple of slides ago that for more than a decade or so, researchers are uploading measurements of their experiments into databases. More than 1 million samples exist in this way. And they not only upload these data, they also actually are enforced to provide metadata for each of their uploaded measurements. So here I show you one example metadata entry for this for one sample. And you can see it has a cryptic title T2-4 and a lot of different fields, raw metadata, and contains subfields, etc. And I can assure you there is a lot of interesting information here that could help us to generate conversations about this measurement, but there's also a lot of trash and redundant information that doesn't tell anything about the biology of the sample. So what we do in reality is we first use, as I said, a language model such as ChatGPT. In our case, we use Mixtral because it's an open source version and therefore much cheaper because we need to process 1 million samples. And we use this to filter this large block of metadata for each individual sample separately. And we use the prompt something like filter biological information and write a short abstract. The real prompt is much larger and we engineered it in an iterative procedure. And what you get out of this, for this one example here, is this very concise biological description. It's still heavily biological. It's renal cell carcinoma tissue sample taken from a male, etc., etc. But this is really describing a lot of biology in very small amount of text, so it's great. And this we then use as basis for a second processing step in which we generate these conversations, these question-answer pairs. And there we have our full system. We have a data set of conversations about individual cell measurements. We have an architecture for a multimodal language model, and we can train it with this. Now, in the next slide, I want to show you a quick demo of this trained AI model and what it can really do for you. And I show this embedded in a graphical user interface to analyze single cells. Now, this might look a little bit confusing if you're not used to this kind of data, but what you see here are 10,000 individual cells. So each dot here is one cell, and for each cell, for each dot, we have the full set of gene activation measurements. So each dot corresponds to 20,000 measured values, essentially. And in the GUI application, what you can do is you can navigate around and explore, and with the power of the AI model, you can now encircle cells that you are interested in. Could also be one cell, and you can just select them and ask questions about them. What that cell is doing, for example. You can ask anything, of course, and also ask follow-up questions, and then the model comes up with a description of the biology of the cell. Thanks. So while this is basically the main part of our research, I would also like to take a couple of minutes to talk about how we can hack cells. And let's start first of all with why we would even want to do that. And there are, I can assure you, a lot of different reasons and very creative reasons for sure. But maybe the most intuitive one can be described with our friend here, Blohai. So Blohai is, we all know him, we all love him. But in our scenario, or no, he's sick. So what do we do with Blohai? We go, of course, to the hospital. And we are really lucky to live in 2024, and for a lot of diseases we have very effective cures. That's great, but still there are also quite a lot of diseases for which we don't have cures, and sometimes also some therapies only work for some people and not for others. So what would it be a great application, for example, for hacking cells, would be if a new patient comes to the hospital, that we kind of take a sample of their cells, analyze it, and then ask an AI system how to specifically alter or modify these cells here to make them healthy cells again. For example, via a medication or a drug that's already known. And there are different ways to get there, and I already showed you, for example, the method of introducing a gene into an organism. This also exists for patients. It's called gene therapy. It's a method that's becoming more and more popular. And I also would like to take here the opportunity to highlight the work of David Baker, who earned the Nobel Prize in Chemistry this year for computational protein design. And in fact, some of the recent methods they're using are also using generative AI approaches to design proteins with the specific functions. And we will see more and more in the future how these kind of generated proteins will also be used more and more in therapeutic applications. However, the kind of research agenda that I'm pursuing is, again, like following the state of cells and trying to understand and manipulate these states of cells, again, using language models. So the idea is that you pass a cell measurement of a diseased cell, for example, or maybe of an immune cell that you want to modify to cure disease, and ask the model how to make this immune cell more active. How can I modify or hack the cell? And the AI can come up with, for example, small molecule drugs, with medications, with different ways to manipulate them and hack them towards a better or curing state. Now, since this is ongoing research, I don't want to go into the specifics of this, but the core principle of the model is very similar to what I was talking about. Again, we rely on public data sets, and this time we train an AI model that, again, understands natural language, but it not only focuses on the states of individual cells, but also on transitions between them. So it tries to understand how a cell transitions from one to another state on a big data scale, and then we can use it to also get suggestions for kind of getting an immune cell into a more active state, or for curing a diseased cell towards a healthy state. All right, with this, I would like to slowly wrap up this talk, first with a small recap. I showed you cells are extremely diverse and extremely complex, and we can use, or AI can help to decode them and help us interpret about their function and identity. And more, we can also leverage these technologies to also understand how we can manipulate these cells for different purposes, including therapy. Now, many people wonder, are there a lot of risks coming from these technologies? Like, will it become super easy to build bioweapons, or could false predictions of AI models lead to harming patients? I think we should take these kind of risks extremely serious, and I'm very glad that there are a lot of researchers actually working on AI safety and considering these kind of risks. Nevertheless, at the same time, I observe that in many discussions, these kind of risks are overproportionally mentioned, I would say, because we see in our field that these kind of technologies already help patients today and will more so in the future. For example, the problem with false predictions, nobody is thinking about replacing any doctor with an AI system today. So usually they are working as assistants and helping with making predictions about diseases. So as of now, I personally don't worry so much about risks, but it's good to keep an eye on it, of course. I would also like to close with a kind of vision about this kind of beautiful hacking and maker scene here. Looking back multiple decades, it became super popular to hack software. This is also one of the roots of this community. And with hardware platforms like Arduino, it became also mainstream to hack hardware. More and more also 3D printers by now are super mainstream and used in many applications. And I hope that in the near and the close future, we will also see more and more biology hacking, maybe at home, maybe at hacker spaces. And of course, there are a lot of questions we need to ask and also to find answers to in terms of feasibility, at risk, security, safety. But at the same time, there's a lot of potential because biology is ultimately about understanding and reverse engineering a super complex system. And I know for a fact that this is really one of the expertises of this crowd here. To give one concrete example, what could already work today and be used in a medium-sized scale is to use sequencers, small sequencing devices that are already in a price range of thousands of dollars. And individuals can and could kind of sequence their blood, sequence their microbiome, like bacteria in your stool, to understand more about your own biology. And with this, I would like to thank all of you again for coming to this talk, and also acknowledge a couple of people with all of them I worked together on the project I was presenting. Especially I would like to highlight Peter Peneder, who is co-first authoring this work with me, and also my advisor, Christoph Bock. There's a project website, so if you're curious to find more, just go there. And if you want to get in touch, here's my website. I'm also on Mastodon, but I didn't put the link yet, sorry. And I'm happy to taking any questions. Thank you.
Thanks a lot, Moritz. Thank you for your talk. So before we start with the Q&As, so you can already queue up in front of the microphones, something special happened. So you got a postcard.
Wow. Thank you so much.
And it's a real cool postcard, so I didn't read it, but I looked at the stamp, and it's a hand-drawn stamp with a DNA strand on it and the ATZG. So
it's very sweet from very sweet people.
Yep. Okay, so are there any questions? I think we start with microphone 1.
Hi, so with volunteer computing projects like Rosetta at home moving to AI from distributed computing, do you still see a way to distribute all of this and take in volunteer work? Or is this all going to move to AWS now?
Yeah, good question. Like Rosetta at home was a project, maybe to provide some context, to make protein folding, like understanding how amino acid sequencing fold into structures, how to make this feasible computationally, because back then this was used in kind of simulation random testing, like Monte Carlo way, and this took a lot of compute. And by now these kind of approaches are largely substituted by AI methods, which just use internal heuristics and work super efficient. And there is actually a large democratization coming along with it, because the AI models that solve this particular problem, they are not immensely large, so many people can run them on their gaming graphic cards. I guess participation is anyways kind of the vision, always requires some kind of knowledge of the field you go into. Biology is extremely complex, so it's good to focus on one thing, let's say, and find your problems there. I hope that AI will also make a lot of things more accessible, especially when it comes to interpreting biological data, because for now, today, we need a lot of bioinformatics, a lot of tool use, understanding a lot of specifics that are actually not really helping in biology and in understanding the biology, rather than only implementing a lot of things. I hope that helps a little bit to get an intuition.
Okay, thank you very much.
Thanks. I would like to ask the Signal Angel, are there questions from the internet?
Yes, there are. One of them, I'd like to know if the manipulation of individual cells is sufficient, or if the interaction of cells isn't more complex.
Yeah, this is a very good question. And of course, there are limitations, that's true. Many types of diseases, disorders are probably more on the whole organism cell, on the whole organism level, or on a tissue level, and can not only be addressed by manipulating individual cells. And I still believe that you can find ways in many circumstances of manipulating cells and thereby manipulating these higher order structures like tissues and whole organisms, but that would require in next steps to not only look at individual cells, but look at whole tissues. So look at multiple cells at once, for example, that make up a tissue. So this is definitely research focus that needs to be addressed more, and is addressed also right now already by AI models as well.
Thanks. Microphone number 6.
Yeah, thanks for the nice talk. I would like to ask an ethical question. So earlier today there was also a talk by somebody from nature preservation, and she was really worried about like balance in nature, because there's all these interactions that we currently don't understand with animals, plants, fungus, and so on. And now there comes into play people that basically exchange genes in these animals, or want to revive animals that are already extinct, and then interact with the system that they don't really understand, right? So the question is a little bit, or I would like you to comment on that.
Well, there's a lot in there. So I know, for example, of this project to bring back a mammoth, and the concerns with it, that it doesn't, first of all, it's impossible to bring it back in the way that it lived back then. It would be a different type of organism, and also it wouldn't have the same habitat as back then. So these are, of course, ethical considerations. I don't dare to comment on them too much, because I don't know so much. When it comes to introducing genes into humans, gene therapy, this is, of course, also like an extreme delicate matter. First of all, it's important to understand that never germline cells, so sperm cells or ovarian cells, are never manipulated in these kind of therapies. So it's always only an individual that's being manipulated. And also these kind of therapies are very much at the beginning, and a lot of ethical considerations are still being discussed as we speak.
Thanks. Number 2.
Thanks. Connecting to the criticism of the false predictions, I was wondering if there is a method, or if you already do this, to measure the uncertainty of your model. Like, for example, you train it on only 80% of the data, and then you run it with the remaining 20%, and you see how many wrong predictions you make, and like that you have a measure of uncertainty.
So generally, as I understand, you're asking about how we can understand how well the model performs. So we go beyond that, and people try to typically go beyond just like doing a train-test split, because then because it's safer to do out of distribution predictions with like independent test sets, basically. So this is what we did. You can read more in the publication on this link. But generally, we use different data sets and try to, for example, predict the types of cells based on their measurements using our models. And we also predict a couple of other cellular attributes.
Do you have a measure of how well you were predicting, like a certainty measure or something?
Like a ballpark number? Yes. It depends so much on what you try to predict. So for specific types of cells like cell types, so there are 200 different cell types that people know of and work with a lot. And for many of them, we have like an accuracy of 80 or 90%. But some cell types are super challenging to predict, for example, because they're super similar to other cells. Also some diseases we can predict very well, but this again depends on the disease. Some diseases are, as we already spoke about, more not so well reflected, not so strongly reflected on individual cells, so it's kind of hard to detect and decode them. For others, it's very easy.
Thanks. Another question from our signal angel, please.
I know biology is not chemistry, but isn't it somehow also problematic to make AI with biology or chemistry? And then there's a link given to an article which references the development of problematic chemical compounds that could be used as chemical weapons.
Yes, so I put this chemical weapons or like bioweapons on the last slide, but didn't speak about it. As said, like there are risks that need to be taken into consideration, and we need to discuss and think about what really will be risks. As we stand right now, I think the limitation is not to design new bioweapons, but rather to synthesize them and to deploy them without being detected. And because even for right now, there are a lot of sequences known for viruses that are super deadly, but people don't create them nevertheless. So it's not that AI or creating new sequences of bioweapons is the real limitation that we face right now. That's one of the stands I have on this.
Thanks. One more question from microphone number 1.
Yeah, thanks again for the talk. I have another question on the precision of the prediction. You said you rely on the training data on mRNA data, basically, if I understood it right. Do you have any data sets where you would include also proteomic data and metabolomic data so that you really accurately determine the state of the cell, not only the mRNA status? So that would maybe increase the precision?
Yeah, maybe some context to this question. Like, there's an alternative to measuring the messenger RNA in your cell, which is like to try to directly measure the proteins available in each individual cell. That is like more accurate because between the step from mRNA to protein, a lot of things can still happen. So mRNA measurements are always only a proxy of the cell state and not really what's really going on the protein level. Now, proteome measurements also have their limitations. For example, you have a tendency to be able to measure fewer individual proteins at the same time. Now, we haven't tried to work with this data. Of course, there's a high correlation between these two, so it would work to some degree for sure. But at the same time, it doesn't make sense to use it as training data probably because the amount of proteomics data is much more limited.
Okay, thanks.
One more question from number 3.
Yes, fantastic talk. I think RNA-seq data have this really big untapped potential, which I'm glad you're addressing with large language models, especially around the concept of cell type, which we've defined as humans so far. So I was curious if your large language model was able to predict new types of cells or get you to move beyond this human concept of a defined box in which cells fit.
Yeah, that's a very good point, and it points also to a very important limitation of this work, which is, of course, that we can only predict and help understand what is already known, because this is what the language model sees in terms of its text input. So it can only reproduce these parts. So for now, this AI is mostly there to help you analyze new data with known concepts and interpret them in a very easygoing manner, let's say.
So one last short question, short answer from number 1.
Are there ideas out there to find out, or using probably ideas of a model to find more results? Like if the model out of the genes make some diseases or indicate something, we could also find something similar, or can we find there new things on, or does it not work? But it probably was answered.
I think one thing I could add to the previous answer is that indeed, if we have a new system, biological system that we are studying and trying to analyze with AI systems, it cannot come up with a creative new word or a new term for these kind of things. But what it will do is it will relate it to known concepts, and it will give you hints what it is comparable to in terms of the biology we already knew. And there it can be very useful and helpful, actually.
Thanks a lot for all the questions, and thanks a lot for the talk, Moritz. So another big applause.